A photon of light strikes a pigment molecule in a light harvesting complex and is relayed to other pigment molecules until it reaches one of the two P680 chlorophyll A molecules in the photosystem 2 reaction center. It excites one of the P680 electrons to a higher energy state. This electron is captured by the primary electron acceptor. An enzyme splits a water molecule into 2 electrons, 2 hydrogen ions, and an oxygen atom. The electrons are supplied one by one to the P680 molecules, each replacing an electron lost to the primary electron acceptor. I think that's it. Photoexcited electron passes from the primary electron acceptor of photosystem 2 to photosystem 1 via an electron transport chain, similar to the electron transport chain that functions in cellular respiration. The electron transfer chain between photosystem 2 and photosystem 1 is made up of the electron carrier PQ, a cytochrome complex, and a protein called PC. The exergonic fall of electrons to a lower energy level provides energy for the synthesis of ATP. Meanwhile, light energy was transferred via a light harvesting complex to the photosystem 1 reaction center, exciting an electron of one of the two P700 chlorophyll A molecules located there. The photoexcited electron was then captured by photosystem 1's primary electron acceptor, creating an electron hole in the P700. The hole is filled by an electron that reaches the bottom of the electron transport chain. Since Calvin cycle requires 3 more ATPs to be inputted into creating the G3P, the cyclic electron flow. I don't know what to say.
Yeah, I mean, you're in.
Since the Calvin cycle requires 3 more ATP, the cyclic electron flow creates more ATP for the Calvin cycle to use, and this only occurs in photosystem 1.
Now that we have talked about light-dependent reactions, we'll talk about light-independent reactions. The first step in the Calvin cycle is carbon fixation. The Calvin cycle incorporates each CO2 molecule one at a time by attaching it to a 5-carbon sugar named ribulose biphosphate, abbreviated as RUBP. The enzyme that catalyzes this first step is called rubisco. The product of the reaction is a 6-carbon intermediate so unstable that it immediately splits in half, forming 2 molecules of 3-phosphoglycerate. The next phase in the Calvin cycle is reduction. Each molecule of 3-phosphoglycerate receives an additional phosphate group from ATP, becoming 1,3-biphosphoglycerate. Next, a pair of electrons donated from NADPH reduces 1,3-biphosphoglycerate to G3P. Specifically, electrons from NADPH reduce the carboxyl group of 3-phosphoglycerate to the aldehyde group of G3P. I think that's all. The next phase in Calvin cycle is the regeneration of the CO2 acceptor, RUBP. In a complex series of reactions, the carbon skeleton of 5 molecules of G3P are rearranged by the last step of the Calvin cycle into 3 molecules of RUBP. To accomplish this, the cycle spends 3 more molecules of ATP. The RUBP is now prepared to receive CO2 again, and the cycle continues. The Calvin cycle is repeated a total of 3 times using 3 CO2 molecules to form 1 G3P molecule, which is a simplified version of glucose.
Thank you for listening. Ow. Oh my